Welcome back to the Learning Studio. This is the Biological Sciences series, and today we're starting a new section, Microbiology and Virology. Our focus in this episode is on bacterial structure, genetics, and antibiotic resistance, a topic that connects microbiology, medicine, and biotechnology in fascinating ways. If you missed our last lecture on biodiversity and conservation biology, the full playlist is linked below in the description. Here's a thought. How can a tiny single-celled organism outsmart the most advanced drugs we've ever created? And why is understanding bacteria not just important for medicine, but also for biotechnology, food production, and even environmental science? Let's begin with their structure. Part 1. Bacterial structure. Bacteria are among the oldest and most successful life forms on Earth. Though small, their structure is efficient. Cell wall provides shape and protection. Gram-positive bacteria have thick peptidoglycan layers, while gram-negative have thinner walls and an outer membrane. Plasma membrane regulates what enters and leaves the cell. Cytoplasm contains ribosomes for protein synthesis and enzymes for metabolism. Nucleoid, a region with circular DNA since bacteria lack a nucleus. Plasmids, small extrachromosomal DNA circles carrying useful genes, often including antibiotic resistance. Flagella and pili help in movement and attachment. This simplicity makes bacteria incredibly adaptable and quick to evolve. Part 2. Bacterial genetics. Bacteria reproduce through binary fission, creating identical clones. But their real power lies in genetic diversity, achieved through mutation. Random changes in DNA often creating new traits. Horizontal gene transfer, HG, sharing genetic material between bacteria via transformation, uptake of DNA from the environment. Conjugation, direct transfer via pili. Transduction, DNA transfer by viruses called bacteriophages. This ability to swap genes makes bacteria like tiny innovators, constantly upgrading their survival toolkit. Part 3. Antibiotic resistance. Now the major challenge, antibiotic resistance. Antibiotics revolutionized medicine, but bacteria fight back through enzyme production, breaking down antibiotics, e.g. beta-lactamases destroying penicillin. Target modification, altering binding sites so drugs no longer work. Efflux pumps, actively pushing drugs out of the cell. Plasmid sharing, spreading resistance genes across bacterial populations. Case studies: MRSA, methicillin-resistant Staphylococcus aureus, resistant to multiple antibiotics causing hospital outbreaks. Multi-drug-resistant tuberculosis, MDR-TB, a global health crisis requiring stronger treatment regimens. Superbugs, bacteria resistant to nearly all available antibiotics, threatening a post-antibiotic era. Why this matters in biotechnology and applied biology. In medicine, understanding bacterial genetics helps develop new antibiotics and therapies. In biotechnology, bacteria are engineered to produce insulin, vitamins, and biofuels. In ecology, bacteria recycle nutrients and even clean up oil spills through bioremediation. Studying bacteria gives us both challenges and opportunities, the fight against resistance, but also the power to harness microbes for human benefit. Past, present, and future relevance. Past, antibiotics like penicillin revolutionized healthcare. Present, antibiotic resistance is one of the greatest global health threats. Future, CRISPR, phage therapy, and synthetic biology may offer new solutions. Why watch this playlist? This playlist covers the 30 most important topics in biological sciences, connecting classical knowledge with cutting-edge applications. Whether you're a student, a teacher, or just a curious learner, this series helps you understand both the science and its real-world impact. So that's our journey into bacterial structure, genetics, and antibiotic resistance, a world where the smallest cells challenge human survival, but also power some of our greatest biotechnological achievements. In our next lecture, we'll continue in microbiology with virus structure and replication, uncovering how these mysterious particles invade cells and change the course of life itself. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the next episode. Because once you understand bacteria, you begin to see both the dangers and the opportunities hidden in the microbial world.